Hello students, welcome to the theory class of Principles of Plant Breeding. Today we'll be talking about emasculation and pollination techniques in important horticulture crop plants, part 2. Earlier we have talked about emasculation and pollination techniques in general, and for some specific crop plants such as tomato and brinjal. We'll be discussing here for some more crop plants which are important in horticulture perspective. So coming to the first one, we have chilies. So in chilies, what we have here is it is considered as self-pollinated crop plants. While it is not highly self-pollinated crop plants, so you can expect some amount of cross-pollination also. So as a general purpose, as general phenomena in self-pollinated crop plants, we have hermaphrodite flowers. So in hermaphrodite flowers, what you can expect is a staminate and pistillate flower are both present in the single flowers. Okay, that means androecium and gynoecium are present in the single flower. Okay, so emasculation is compulsory here because we cannot wait for the plants to flower. We cannot wait for the flower to anthesis and dehisce and get self-fertilized. Okay, what our intention should be in terms of hybridization program or in terms of crossing program, we have to look out for either a staminate plant and a pistillate plant. Okay, so in order to make a hermaphrodite flower, which is where androecium and gynoecium are both present, we have to remove androecium to make the flower a female pistillate flower. Okay, so it can be done. We have already discussed about what is emasculation. It is the process of removal of anthers, which are the source of pollen grains. So if it is not done so the pollen grains will dehisce inside the closed flower and the fertilization will take place, and there will be self fruit in spite the cross-pollinated fruit that we are expecting. Okay, so what we have to do, remove the anthers and according to emasculation process. So in terms of chilies, uh, we have to select the right stage of buds, the right stage of flower buds, and the right stage of flower buds is 1 to 2 days before the flowering. Okay, so how you will be able to identify this? It comes with it comes with the experience and art, or you can call it as a skill of the plant breeder, or if you're employing JRF research fellow, or you can say a farmer which is willing to go for seed production, so he or she might be able to identify this stage of flower buds in order to make the flower buds open in the next morning. Okay, because if you select immature flower buds, what will happen, your emasculation process will be infertile because it will take 1 week or so to mature and to start the stigma receptivity. Okay, as soon as the anthesis starts, that means the flower opening starts, or you can say the anther dehiscence, pollen dehiscence starts, the stigma receptivity also starts. So you have to look at the time period between the emasculation and the stigma receptivity. Okay, so first one we have here is the removal of, in case of chilies, what we have to do is removal of the petals, and along with that the anthers which are surrounding the stigmatic uh, the stigma. Okay, and you can call it as a style. Okay, so once you have done this process, you have to bag it, as it is the most important part, because as we were talking about the amount of cross pollination that may happen in chilies. It is not highly self-pollinated, so you can expect a little amount of cross-pollination from a nearby variety. Okay, so you can bag it, okay, with a colored bag, or you can bag it with a simple brown paper bag. Okay, so that it is protected against the environmental fluctuations or weather conditions, or even from the pollen grains, which can cause genetic admixtures. Okay, Coming to the next slide we have here is the hands and tools which are going to be used in the crossing program, emasculation and pollination program. Okay, so the forceps, here we have forceps, needles with handle, and of course the scissors. And the most important thing here is sterilization of these equipments. Okay, first thing you can do here is at the most you can sterilize it in oven. Okay, what we normally do in biotechnological lab is sterilize in oven, and you can also sterilize this in autoclave, or if you're not doing that, you can at least do it with the ethanol. Okay, 70% ethanol acts as a sterilant. Okay, so once you have done this, you can use this for removal emasculation process. Okay, forceps is normally used for removal of anthers, which is easy to pick up. Needles are also sometimes used when the flower is quite small, and of course they are used, needles are used to open the flower buds sometimes. Okay, and scissors are usually used to cut delicate flower buds. Okay, so these are important, like flower petals. Okay, so these are important equipments which is inevitable for a plant breeder. Of course there are others like lens, and because we are not going to sterilize the lens, I haven't included that equipment here. The next thing is you'll be needing clothes. Okay, some cheesecloths and some lightweight cotton strings, and you can also use cotton swab, or you can also use cotton threads. Okay, threads of different colors, tags. Okay, and you can also use brown paper bags, okay, of different colors, so that you can cover it after emasculation process. Okay. Okay, next thing is pollination, pollinated flowers. Okay, once you have done the emasculation process, in the next morning, say for example from 8 o'clock to 9 to 10 o'clock, so you can pollinate it, because in that hours 8 o'clock to 10 a.m. in the morning, so the flower anthesis will start in the male lines. Okay, from the male lines you are going to collect the pollen grains which are yellowish. Okay, make sure that the pollen grains are yellowish. If it is quite whitish in color, it indicates that it is over matured and it won't fertilize the emasculated stigma. Okay, so you have to make sure that the uh, the pollen grains are white in color and it is bright. Okay, so it is also it comes under the skill of the plant breeder to observe this pollen grain color, and at the same time the amount of pollen contained in the male pollen shader. Okay, so the pollen shader will be grown in a different plot. Okay, and the emasculated flower will be grown in a different plot. Okay, or sometimes it is grown in a single row. Okay, one alternate rows. Okay, and when you select this, you have to initially identify the male line which you are supposed to get pollen grains from by tapping the flower. Okay, so once you tap the flower, if there is a powdery substance oozed out from the anthers, or you can say the flower itself, so you can expect uh, viable, or you can say uh, promising pollen grains. You can collect a huge amount of pollen grains as much as you can, and you can pollinate in most of the emasculated flowers. Okay, and right after emasculation, what you have to do is to bag it. Okay, right, and pollen grains is transferred into the stigma either from the matured anthers by scooping it out through the lateral sutures with the needle, or by touching a freshly dehisced anthers to the stigma with a forceps. Okay, so there are different methods with which you can pollinate. First, I have indicated here with the help of cotton swab, or you can also use either brush, okay, or painting brush, or you can use forceps, or you can collect it in a petri plate, and then you can finally and smear it into the stigmatic surface. Or what you can do is you can collect the male flower and you can directly smear it. Okay, so these are the things you have to take care of. And the next thing is tagging is important because in order to identify the pollinated flower from the unpollinated flower, say for example, if it is immature, if you don't want to emasculate, if you don't want to pollinate today because the stigma receptivity is not there, so you will have to the flower buds will have to wait for 1 or 2 days. Okay, so you can untag it and you can take those flower buds which are you are going to pollinate or you have already pollinated. Okay, so in this way you can identify those flowers where the crosses can be expected. Okay, and in future if you don't uh, cross this pollinate this one, so it will yield no fruits. Okay, pollinated flowers are protected from bees by double layers of cheesecloths loosely wrapped around the branch, enclosing leaves and flowers and securely fastened. So you know now that the importance of cotton cloths, or you can say butter paper, because all kinds of pollinating mechanisms are there, entomophily, okay, anemophily, and there are lots of bird pollination. Okay, so in this case what you have to take care of is pollination by cross pollination by insects. Okay, and of course in highly cross pollinated crop plants there is huge amount of and anemophily, that is pollination by wind. Okay, and the next thing is approximately mark plastic labels describing the cross, the date, and attached to the bamboo stake marking the chosen plant. Okay, so clearly you have to mark the plant also. Okay, that means say for example when you are planting when you are emasculating a group of lines, you can tag the line nearby that line. Okay, at the beginning of the line, so you can tag you can insert a peg into the land, okay, as a landmark to indicate that these lines are the cross lines where you are expecting the hybrid variety. Okay, and of course if you're not going to do in a single line, or if you want to do in an individual plant, you can also indicate by keeping a tag along with the peg in the nearby plant that you have emasculated and pollinated. Okay, the next thing is pollinated flowers are periodically checked and the cloth is removed for 46 days. Fruits normally mature in about 45 days. We are going to remove the uh, butter paper or cheese bag right after 4 to 6 days, or you can say a week, because you have to uh, expose the fruit, the growing fruit, and then you can say the hybrid fruit into the external environment so that it gets hardy. Okay, the next thing is next crop plant is Bhendi. So in case of Bhendi, the type of pollination mechanism is often cross pollinated crop. So what you can expect is the since there is self-pollinated mechanism, what you can expect is the hermaphrodite structures of the flower. Okay, but because of the flower opens during flower opening during the anthesis process, that means flower pollen dehiscent stage. So it starts, some amount of cross pollination may happen during those time. Okay, and emasculation in this process are done with selecting buds which are likely to open in the next day and are given a slight ring cut at the base of the flower, but with the help of a blade. Okay, so you can expect a ring cut. Okay, and you can break open the calyx, what you can say, the outermost cover of the flower. Okay, so right inside it you can see the white color petals or corolla, and petals along with calyx sheets are removed and staminal columns are and the stigma are exposed. Okay, so you remove the petals and calyx and it is the staminal column, that means the pistillate column is exposed. Okay, so in this way, right after the emasculation, that is after ring cutting and removal of the corolla and calyx, you can cover it with a brown paper bag, or you can color it with a colored paper so that it is protected. And the next thing is um, in the next morning the bags are removed and the emasculated birds are pollinated. Okay, and what kind of flowers we have to select for male from the male plants? We have to select those newly open flower and with yellowish color or bright color, pale bright color anthers, or you can say the pollen grains in the in the flower. Okay, so you can directly pluck this flower and emasculate the pollinate in the stigmatic surface. Okay, stigmatic surface. So once enough amount of pollen grains are attached on the stigmatic surface, you can expect some amount of process in the future. Okay, so, and of course, not only this method, you can also use pollination method with the help of the brush. Okay, so both methods are effective. You can use either ways. The next plant is garden pea. So it is the most talked about, and it is the plant which has been done research in terms of genetics point of view, the most highly researched crop plants apart from maize. Okay, and maize, and another thing is Arabidopsis thaliana. Okay, so when it comes to cultivated crop plants, it is maize and green pea. Okay, especially green pea in case of genetics. Okay, because this is the crop plant where Mendel discovered the two basic laws of inheritance. Okay, what you all know. The first thing is emasculation. Again, the flower buds. The same thing here. You have to select the young flower buds which are supposed to open in the next 1 or 2 days. Again, next and after that flower opening. So you can see here, understanding the flower morphology is quite important before you initiate the emasculation and pollination program. Okay, because first of all you have to know whether the flower is a hermaphrodite flower, and the next thing is, if it is a hermaphrodite flower, what kind of petals are there. Okay, if the anthers are attached with the petal, or if it is detached with the petal. Okay, if the anthers is attached with the stigma. Okay, all kinds of stuff are there, all kinds of morphological differences are there, so you have to understand so that you emasculated it successfully and you don't destroy the flowers during the emasculation process. Okay, so this is the most important thing. Basically, in case of cross-pollinated crop plant, what you can expect is the staminate and pistillate flowers are present in two different plants or two different parts of the plants. Okay, so these are some important things. So in case of peas, in case of pulses, or you can say in case of legumes, the typical flower structure is this one. There is the outermost petal, okay, which is called as banner, okay, which acts like a flag. Okay, and the next thing is a wing which covers the keel petal. Okay, wing petal, banner, wing petal, and keel petal, which is the last one. So in case of emasculation process in legume crops, what people normally do is remove the keel petal only, or remove all these wings. Okay, banner, wings, and keel petal. Okay, you can directly remove keel petal, and sometimes during when you remove the keel petal, okay, in some legume crops the anthers are attached with the keel petal. So when you pull out this keel petal with the help of a forceps, all the anthers automatically comes out of the flowers. Okay, so the emasculation process is quite easy in case of some legume crop plants. Okay, say for example groundnut, you can directly pull the keel petal with the help of a forceps. Okay, with the help of a forceps, so anthers, and since anthers and anthers only, the stigma is detached. Okay, anther is attached to the keel petal, so if you pull out the keel petal, so the anthers are all removed. Okay, and after emasculation process you have to make sure that any anthers which is left inside uh, the flower. Okay, so flowers can be emasculated anytime. So emasculation is recommended anytime in this process. Normally, in case of most of the crop plants, emasculation is done in the evening, as we have already discussed. The first step in emasculation is to tear away with the forceps the tip of the sepals. Sepal means what? The calyx, the singular form of calyx. Okay, and in front of the keel, and in front of the keel. Okay, and the forefinger is positioned behind the flower, and the thumb in front, and a light pressure is applied. Okay, so here you have to apply a light pressure, and this spreads the standard and wing to expose the keel petal. Okay, so when you press a light pressure here, so the wing and keel petal is exposed, that means it is opened. Okay, and then next thing is the exposed keel petal is slit open by tip of the forceps, and a pressure can be applied by the thumb and fingers on the keel for increased exposure of the pistil and stamen. Okay, so when you apply this, the keel petal will be open, and the 10 stamens can be pulled out of it. Okay, so this is the method of emasculation without plucking out, or without destroying the corolla and calyx. Okay, most of the corolla and calyx, because the main advantage of this is the protective cover remains attached with the in the emasculated flower. Okay, and make sure that the flower is tagged with a color tag. Okay, color thread, in order to identify, because in this case it's if you leave like this, it will be difficult to identify which is emasculated flower and unemasculated flower. Okay, so these are the basic steps here. Remove the outer petal, remove the keel petal, expose anther and stigma, remove all the anthers. Okay, these are the basic steps of emasculation. And the next thing you have to do, you all know, the pollination process. Okay, pollen can be obtained throughout the day. Okay, so the emasculation and pollination process of uh, bee. Okay, it is quite easy. It is quite easy. That's why this is one of the factor why Mendel chose and Mendel got succeeded. Okay, because it is, you can, in this case, you can emasculate at any time, you can pollinate at any time, because the stigma receptivity and anther dehiscence is all throughout the daytime. Okay, so but you have to make sure the flower opening of both the desired male plant and desired female plant that you are supposed to cross are at the coincident time. Okay, because the receptivity and stigma receptivity and antithesis works on the same time. Okay, if you skipped, you have to if say for example, in case of the male plant that you are supposed to use, okay, that you are supposed to collect pollens is a late maturing, that means a late flowering plant. Okay, so you have to plant it earlier than the female flower. Okay, and the female flower can be planted a little bit later. Okay, so that the flowering time of both the male line and the female line is matched. Okay. This type of breeding is called staggered breeding. Okay, staggered breeding means matching up the flowering time of both, so that crossing program is possible. Okay, and for pollen collection, it is more convenient to pick the male flowers and remove the standard and wing petals, and pull back the keel so that the style protrudes, and use the pollen covered styler brush as an applicator to transfer the pollen to the stigma of the emasculated flower. Okay, so all kinds of methods are given here. uh, so all the process of all the method of pollination can be done, like with the use of brush, with the use of, you can say, what flower, direct flower pollination, or you can collect in a petri plate and then directly apply there. Okay, all flowers and other flowers are not used in crossing and are removed to peduncle to increase the pod shed after crossing. Okay, so So once you have emasculated or once you have pollinated, okay, you remove other crop, other flowers. Okay, normally in case of cultivated uh, plot, or you can say cultivated crop plants, we are not supposed to remove the flowers because these are the source of the pods or the fruits that is supposed to, you all know, fruit is originated from flowers. If you pluck out flowers or if the flower drops, it will be a loss to the farmer. Okay, and but in case of this seed production, it'll be quite wise to remove the unemasculated flower or unpollinated flowers. Okay, because in order to avoid much of the confusions which can be created due to unemasculated flowers. Okay, and there may be genetic admixtures while harvesting also. Okay, because we are not supposed to, a plant breeder or a scientist is not supposed to not usually don't harvest. Okay, so that's why during harvesting some farmers may mix it up. Okay, so you can at least ensure the genetic purity with by plucking out the unemasculated flower, or you can say unpollinated flower during seed production, hybrid seed production program. Okay, so this is the end of the slide. If you have any doubts, I'll be eager to help. Thank you.